Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, today we are going to start with excretory system of human. In the previous lecture, we have introduced excretory system. We have covered general concepts of excretory system, and now we are starting with excretory system of humans. Humans or mammalian urinary system. It consists of a pair of kidneys, a pair of ureter, urinary bladder, and urethra. It comprises of a pair of kidneys, a pair of ureters, urinary bladder, and a urethra. Kidney. Kidney, they are dark, red, bean-shaped organs. The dimensions are 11 centimeter long, 5 centimeter wide, and 3 centimeter thick. So kidney, they are a pair of kidneys which are dark red, bean-shaped, okay, organs of 11 centimeter long wide, 11 centimeter long, 5 centimeter wide, and 3 centimeter thick. Each kidney weighs about 150 gram in an adult male and about 135 gram in an adult female. Okay. They are placed against the back wall of abdominal cavity just below the diaphragm. On either side, opposite the last thoracic and first three lumbar vertebrae. The position, the location is that they are placed against the back wall of abdominal cavity just below the diaphragm on either side opposite to the last thoracic vertebrae T12 to L3. First three lumbar vertebrae. The 11th and 12th pair of ribs protect them. 11th and 12th pair of ribs protect them. Okay. These 11th and 12th pair of ribs, they are floating ribs. The floating ribs, they protect the kidneys. Okay. This is the human excretory system. Okay. I will be coming on this diagram. The kidneys are covered by peritoneum. They are covered by peritoneum on the front, that is ventral side only. Thus, they are retroperitoneal. They are retroperitoneal. The right kidney is attached more anteriorly than the left in rabbit. This asymmetry is just a reverse of that found in man. The right kidney is attached more anteriorly in case of rabbit, while in man, the left kidney is attached more anteriorly than the right. Okay. The concave side of each kidney faces inwards and convex side outwards. Okay. In man, left kidney occurs at a slightly higher level than the right one because the right side has prominent right liver lobe. In rabbit, the condition is little different due to quadrupedalism, that is, left kidney is in normal position while the right kidney shift ahead to provide place for stomach below it. So in man, left kidney occurs at slightly higher level than the right one because right kidney has prominent right liver lobe. In rabbit, the condition is little different due to quadrupedalism. That is, left kidney is in normal position while the right kidney ahead is shifted ahead to provide place for stomach below it. Okay. So please remember these differentiation points that first that the kidney is retroperitoneal because it is only covered by peritoneum on the ventral side, front side, right? The right kidney is attached more anteriorly, okay, in comparison to the left kidney in rabbit, while in man, left kidney is attached more anteriorly, okay. This asymmetry, yeah. Now, in men, left kidney is it is at the higher level. Left kidney is at the higher level than the right kidney because right kidney is suppressed or it is having the right liver lobe on the top of it. In rabbit, the condition is different due to quadripedalism. Left kidney is in normal position, but the right kidney is shifted forward, ahead, okay, to provide place for stomach below it. Okay. In mammals, the kidney is concavo-convex. The center of the concave inner surface is called hilum or hilus, which gives out a ureter. From this hilus surface, the renal artery enters into the kidney, the renal vein comes out, and the renal nerves enter into the kidney. In mammals, kidneys are concavo-convex. The center of the concave's inner surface is called hilum or hilus, which gives out a ureter. From this hilus surface, the artery enters into the kidney, renal vein comes out, and the renal nerves enters the kidney. Okay. Now you can see the picture here. Okay. This is the concave side. This is the concave side. This is the convex side. Okay. So this part is basically known as what? Renal hilum. This renal hilum is basically having, you can see the renal vein coming out, renal artery getting in. Okay. So this is the structure. It is concave or convex. Concave side of each kidney face inwards. Okay. Human kidneys are metanephric. So a pair of kidney, a pair of ureter, urinary bladder, and urethra. This is the structure. Structure of kidney. Kidneys are metanephric in animal mammals. The kidney is divisible into two parts. The outer part is cortex and the inner part is medulla. In humans, in mammals, kidney is metanephric. It is divisible into two parts. The outer part is cortex and the inner part is medulla. Okay. Three layers of tissue surround each kidney. So kidney is covered by a layer of fibrous connective tissue, the renal capsule, which protects it from infection and injuries. Okay. So the innermost layer is renal capsule made up of fibrous connective tissue, which protects from renal injuries and infection. Okay. The intermediate layer, which is made up of adipose capsule, or which is known as what? Adipose capsule, made up of fatty tissue. Okay. So this adipose capsule, which is made up of fatty tissue, it basically act as a thermoregulator to maintain homeostasis. Okay, and shock absorber. The outermost is renal fascia, which is made up of dense connective tissue. Okay. The fascia anchors the kidney to the abdominal wall. Understood? Just inner to the renal capsule. Renal cortex is there. Okay. And inner renal medulla. Okay. This is the longitudinal section of kidney. You can see I mentioned different parts. Okay. The renal medulla, you can see the renal medulla is divided into numerous medullary pyramids, around 15 to 16 medullary pyramids, okay, having a broad base towards the cortex, okay. Now, coming to the labelings of this longitudinal section, the first is renal pyramid, okay. These are renal pyramids. These renal pyramids are basically conical projections of renal medulla. Okay, so these renal pyramids they are known as what? You know, the medulla is basically divided into number of conical areas, and these conical areas are known as what? Medullary pyramids or renal pyramids. Okay, so these renal pyramids around 15 to 16, they have a broad base at the side of cortex. Okay, and all these medullary pyramids it basically terminate into a structure known as renal papilla. So it terminate into a structure known as renal papilla. So number 16, this is a renal papilla. Okay, this is renal papilla. Okay, renal. So renal pyramid, renal papilla. Then efferent vessel, which is basically, coming out. Okay, and which is coming out. The efferent vessel. This is third. Efferent vessel, third. Renal artery. Second is efferent, sorry. Third is renal artery. Third is renal artery. Second is efferent vessel. Second is efferent vessel. This is the efferent vessel. Okay, artery. Renal vein. Fourth is renal vein. Fifth is renal hilum. This whole process through which the renal artery is going in, renal vein is coming out, renal nerve is going in, known as what? Renal hilum. Okay. Sixth, renal pelvis. Sixth, this part is known as what? Renal pelvis. Okay. Seventh is ureter. This is ureter. Eighth is minor calyx. Okay. The part eighth. This part is known as what? This part is known as what? Minor calyx. Minor calyx. Okay. Ninth is renal capsule. Okay. This outer covering is known as what? Renal capsule. Tenth, inferior renal capsule. This is inferior renal capsule. Okay. 11th, superior renal capsule. This is superior renal capsule. 12th, afferent vessel. This blue one is afferent vessel. Afferent vessel. Okay. 13th, nephron. This is nephron. 14th, minor calyx. Minor calyx. 15th is major calyx. Major calyx. Minor calyx merges into major calyx. 16th is renal papilla. Renal papilla already done. 17th is renal column of Bertini. These are renal column of Bertini. So this is the longitudinal section of kidney. Okay, you have to go through these labelings. Okay, once again, so that you can, you know, get it more properly done. Okay. So basically, what happens happening? The medulla is divided into medullary pyramids. Medullary pyramids are merging into renal papilla. Between the medullary pyramids, the substance of the cortex, okay, the cortex extends between the medullary pyramids, which is known as what? Renal column of Bertini. So, what is renal column of Bertini? I'll explain again. It is the extension of cortex in the medulla. So, the extension of cortex in the medullary region basically makes the renal column of Bertini. The medullary pyramids are connected with minor calyxes. The medullary pyramids, they are connected with minor calyx. Okay. Are connected with minor calyx. Okay. Around 7 to 13 minor calyx. The minor calyx lead into major calyx. Two to three in number. Okay. These major calyxes open into a funnel-shaped structure called renal pelvis. Renal pelvis. And renal pelvis opens into ureter. Okay. So the renal pyramids or the medullary pyramids. The medulla is subdivided into 8 to 18 conical masses known as renal pyramid, each having a broad base towards the cortex and a narrow end called renal papillae towards the pelvis. Path of urinary drainage. Collecting duct. The waste goes from collecting duct to papillary duct in renal pyramid. From papillary duct in renal pyramid, it goes to minor calyx. From minor calyx to major calyx. From major calyx to renal pelvis. From renal pelvis to ureter. From ureter to urinary bladder. So the urine, it goes from collecting duct to papillary duct to minor calyx, then major calyx, then renal pelvis, then ureter, and then urinary bladder. Renal columns of Bertini between the pyramids, the cortex extends into the medulla or renal columns of Bertini. Okay, in the diagram, it is like this. This is the extension of the cortex. Okay, you can see the extension of the cortex, this boundary. So this is known as what? Renal column of Bertini. Each renal papilla projects into the cavity of minor calyx. Minor calyx join to form a major calyx. Major calyx opens into a white funnel-like structure pelvis. The later leads the ureter. In rabbit, the pelvis is unbranched, hence it is without calyx. So in rapid, calyx is absent. Okay. In frog, ventral surface of each kidney has many ciliated funnels called nephrostomes. They drain waste from body cavity, coelom, that is coelom, and connect to renal veins in frog or to urinary ferrous tubules in tadpoles. In frog, the ventral surface of each kidney has many ciliated funnels, and these ciliated funnels are called nephrostomes. So in frog, nephrostomes are present. Okay. They drain waste from body cavity, that is coelom. Body cavity is known as what, coelom, and connect to renal veins. And all these funnels, they are connected to the renal veins in frog or to urinary ferrous tubules in tadpoles. Now comes the histology of kidney. Okay, histology is the study of tissue. So histologically, a kidney is made up of innumerable thin, long, much convoluted tubular units called urinary ferrous tubule or nephron. Okay, so around 10 lakh structural function units called nephrons. Okay, so histologically, a kidney is made up of innumerable thin, long, much convoluted tubular units called urinary ferrous tubules or nephrons. Nephron is the structural functional unit of the kidney. One human kidney may contain about 1 million, that is 10 lakh nephron. Okay. In rabbit, each kidney bears about 2 lakh nephron. In frog, each kidney bears about 2000 nephrons. Okay. So, one human kidney may contain about 1 million, that is 10 lakh nephron. Okay. The rabbit, each kidney bears 2 lakh nephron, and frog 2000 nephrons. Okay. Structure of nephron. Nephrons, they are also known as urinary tubule. It consists of malphigian corpuscle and renal tubule. So a nephron or urinary tubule consists of two parts. One is the malphigian body or malphigian corpuscle or renal corpuscle. Okay. The malphigian body or malphigian corpuscle or renal corpuscle. Okay. The proximal end of each nephron forms a blind or closed, enlarged and double-walled cup, the Bowman's capsule in the cortex. Name Bowman's capsule is based on English physiologist and histologist William Bowman. Okay, so the first comes Malphigian corpuscle or Malphigian body or renal corpuscle. The proximal end of each nephron forms a blind. It is not connected. It is open. It is not open. It is blind. Okay. It is a blind or closed. Okay. Enlarged and double-walled cup. Double-walled cup called Bowman's capsule. It is present in the cortex. Okay. Each capsule contains a network of blood capillaries. So each capsule basically it consists of network of blood capillaries, stuffed up blood capillaries known as glomerulus, which receives the blood through afferent arteriole and the blood comes out through the efferent arteriole. The diameter of efferent arteriole is comparatively lesser. Bowman's capsule and glomerulus receives about 20 to 25% of the cardiac output blood at rest. Okay, so Bowman's capsule, also known as the Malphigian body or renal corpuscle. Okay, each capsule contains a network of blood capillaries. Okay, the glomerulus which receives blood through afferent arteriole and the blood comes out through efferent arteriole. The diameter of the efferent arteriole is comparatively lesser. Okay, Bowman's capsule and glomerulus receives around 20 to 25% of the cardiac output blood at rest. The composite structure of Bowman's capsule and glomerulus is known as Malpighian body, known as what? Malpighian body. So the Malpighian corpuscle or renal corpuscle, okay, it comprises of glomerulus and Bowman's capsule. Okay. The composite structure of Bowman's capsule and glomerulus is known as Malpighian body or Malpighian corpuscle after the Italian microscopist Marsilio Malpighi. Okay. So confusing terms that what should we name it? Malpighian body or the Malpighian corpuscle or renal corpuscle, okay, or Bowman's capsule. So Bowman's capsule and glomerulus, these two things when they merge, they form Malpighian body or Malpighian corpuscle, okay. So please remember, I'll write it here. Bowman's capsule plus glomerulus, they form Malpighian body or Malpighian corpuscle. Okay. Then the second part after the Malpighian corpuscle or renal corpuscle is tubule. A tubule is differentiated into three parts: proximal convoluted tubule, Henle's loop, loop of Henle, and distal convoluted tubule. The Bowman's capsule opens into the proximal convoluted tubule. The anterior part of the PCT is more coiled, whereas its posterior part is almost straight. The anterior part is more coiled and the posterior part is almost straight. The proximal convoluted tubule opens into loop of Henle. It opens into loop of Henle. The Henle's loop is a U-shaped structure or makes hairpin turn, which has a distinct descending limb and ascending limb. The ascending limb opens into a distal convoluted tubule, the DCT, which is a coiled structure. Many DCT unit to form a collecting duct. The collecting duct of one pyramid unit to form a duct of Bellini. Okay. The collecting ducts of one pyramid unit to form a duct of Bellini. The duct of Bellini lead into the pelvis part. Okay. So the second part is tubule. Tubule is differentiated into three parts: proximal convoluted tubule, loop of Henle, and distal convoluted tubule. The Bowman's capsule opens into a proximal convoluted tubule (PCT). The anterior part of PCT is more coiled. It is more coiled, whereas its posterior part is almost straight. The PCT opens into loop of Henle. Okay. The loop of Henle is a U-shaped structure which makes a hairpin turn, which has a distinct descending limb and ascending limb. Okay, a distinct descending limb and ascending limb. The ascending limb opens into the distal convoluted tubule. The DCT is a coiled structure. The DCT is a coiled structure. Many DCT distal convoluted tubule unit to form a collecting duct. Okay, and these collecting ducts of one pyramid they unite to form a duct of Bellini, and the duct of Bellini open into the pelvis part. Okay. So first, further discussing, I would like to show the structure of Malpighian corpuscle. Okay, once. So I need a slide to be added here. Just a minute. Hmm, here I should add on. Wait, just a minute. Yeah, yeah. Now I can make the diagram. Okay. So Yes. Now see, there are some specific cells. These are the cells of proximal columnar tubule having brush border epithelium, having microvilli. Okay, you can see the microvilli. The afferent arteriole comes in. Okay, there are specific cells which are podocytes. Okay, they are arranged here. Podocytes mean cells having food processes. Okay, I'm just giving you a glimpse. Okay, rest you can go through the pictures given on the Google. These are podocytes. Okay, this is afferent arteriole. Okay, now this afferent arteriole, it comes and it divides into a tuft of capillaries. Okay, so it divides into a tuft of capillaries. Many of the capillaries they come, they merge to make like these tuft of capillaries. Okay, okay, and it goes out. This is efferent artery. Efferent artery. Okay, you can see the diameter of efferent arteriole is less. These are the capillaries. Okay, so all these are capillaries. Tuft of capillaries. So we have podocytes. These are the capillaries. Okay, this layer is known as what the Bowman's capsule or the glomerular capsule. It is a double-layered cup-shaped structure having a parietal layer and a visceral layer. So this layer is known as what? The parietal layer. This is parietal layer. Okay, understood? And these parietal layer cells, okay, then they are squamous, made up of squamous epithelium. Then comes the visceral layer. This is the visceral layer. This is the visceral layer. Okay. This is the parietal layer. This is the visceral layer. Okay. So this is the Malpighian corpuscle or the Bowman's capsule. Okay. So Malpighian corpuscle, basically the Bowman's capsule along with glomerulus. Okay, that is known as what the Malpighian corpuscle or the Malpighian body. Okay. Now I'll write it here. Parietal layer is outer and visceral layer is inner. The parietal layer consists of squamous cells and the visceral layer surrounds the glomerulus and is composed of special type of cells of podocytes. Visceral layer, podocytes, and parietal layer, squamous cells. Okay, understood? Now, the arrangement of nephrons, the Malpighian body, and the most of the proximal convoluted tubule and distal convoluted tubule. They are situated in the cortex. They are basically situated in the cortex region. Henle loop and collecting duct are found in medulla. Okay, the loop of Henle, okay, and the collecting duct, they are found in medulla, while PCT, DCT, and cortex, they are found, PCT, DCT, they are situated in the cortex, okay, along with Malpighian body. So, Malpighian body plus PCT plus DCT, they are found in the cortical region. Vasa recta. The efferent arteriole of the juxtaglomerular nephrons forms a peritubular capillary system. It forms a very important peritubular capillary system around the loop of Henle, which is called vasa recta. Each of the vasa recta makes a U-turn at the innermost part of the medulla and return to the venous circulation near the junction of medulla and cortex. The efferent arteriole and peritubular capillaries technically constitute a renal portal system. They make a renal portal system, peritubular arteries and the efferent arteriole. In all amniotes, okay, having the amniotic membranes like reptiles, birds, and mammals, they have this renal portal system of efferent arteriole and peritubular capillaries. So, what is vasa recta? Vasa recta is basically the efferent arterioles of juxtaglomerular nephron. They forms a peritubular capillary system around the loop of Henle, which is called vasa recta. Each vasa recta makes a U-turn at the innermost part of the medulla. It makes a U-turn at the innermost part of the medulla and return to the venous circulation near the junction of medulla and cortex. Okay. The efferent arteriole and peritubular capillaries technically constitute a renal portal system. In all amniotes, as reptiles, birds, and mammals, they have a renal portal system made up of efferent arteriole and peritubular capillaries. So these are the types of nephron. Yeah. One more important thing I just want to remember the structure of glomerular membrane. When I'll make you a study about the histology, the functional structure of glomerular membrane is very important. Okay, so the functional when we talk about the functional structure of glomerular membrane, it basically consists of three layers. It consists of three layers. The first layer is endothelium. Okay, this endothelium is fenestrated as it is perforated by small holes. The fenestrated endothelium, known as fenestrae, having perforations. The second is the basement membrane. Basement membrane, okay, which is present outside the endothelium. Outside the endothelium. This basement membrane is mainly composed of collagen and proteoglycan fibers, okay, having large spaces. It is made up of proteoglycan and collagen fibers. Okay, and the third is epithelium. Epithelium, a visceral layer having podocytes. And the third layer is having podocytes. Okay, these podocytes, they are foot cells. Okay, the podocytes, they have foot-like process or projections known as pedicels. Okay, podocytes known as pedicel. Space between pedicel is called slit. These slits or slit pores are filtration slits. Okay, filtration membrane. These slit pores known as are act as they act as filtration membranes. Now, types of nephrons. Nephrons are of two types. One is the cortical nephrons, which are basically in the cortical region, and the other one are juxtamedullary nephrons, with regard to their location in the kidney. The cortical nephrons form about 80% to 85% of the total nephrons. They lie in the renal cortex and have very short loops of Henle that extend only little into the medulla. So nephrons are of two types. One is the cortical nephron, okay, and the other one is juxtamedullary nephrons. The cortical nephrons, around 80 to 85% of nephrons, total nephrons are cortical nephrons. Okay. These cortical nephrons, they mainly lie in the region of renal cortex. Okay, their glomeruli are found in the outer cortex, and the loops of Henle are short and extended into a little extend into the medulla. They don't have vasa recta. Okay, so very important that they don't have vasa recta. Okay, the remaining 15 to 20% are juxtamedullary nephrons having the Bowman's capsule close to the juxta, means the junction of cortex and medulla, and have a very long loops of Henle extending deep into the medulla. This type of nephron is present in only birds and mammals. The cortical nephrons control the plasma volume when water supply is normal. The cortical nephrons control the volume of plasma when water supply is normal. The juxtamedullary nephrons regulate the plasma volume when water supply is short. Okay, in adverse condition. Okay, so the nephrons are of two types: juxtamedullary nephrons and the cortical nephrons. Okay, cortical nephrons they don't have vasa recta. Juxtamedullary nephrons they have vasa recta. Okay, and they control plasma volume when water supply is short in adverse condition. Okay, is it clear? The cortical nephrons they control the volume of plasma when water supply is normal. Okay. The juxtamedullary nephrons they are larger in size. Cortical nephrons they are smaller in size. Loop of Henle juxtamedullary nephrons are very long. Okay. Cortical nephrons, juxtamedullary nephrons forms 80 to 85% of the total nephrons. Forms only 15 to 20% of the total nephrons. Are small in size, are large in size. Juxtamedullary nephrons are large in size. Cortical nephrons they lie mainly in the renal cortex. Juxtamedullary nephrons they have Bowman's capsule in the cortex near its junction with medulla. Okay. In cortical nephrons, the loop of Henle are very short and extend only a little into the medulla. Loop of Henle are very long, and because of the length of loop of Henle, the maximum reabsorption of water takes place. They control plasma volume when water supply is normal. They control plasma volume when water supply is short. Glomeruli is in superficial region of the cortex, outer cortex. Glomeruli is deep cortex, in deep cortex. Blood supply from peritubular capillaries only. Okay. Blood supply from peritubular capillaries and vasa recta. Okay. Histology of nephron. First, we will study about glomerulus. Glomerulus is a network of around 50 parallel branching and anastomosic capillaries which are covered by endothelium. As I have given you the names, the outer is endothelium, the middle is basement membrane, and the inner one is epithelium made up of podocytes, which has slit pores or filtration pores that restrict the passage of the colloids. Okay. However, small molecules and water can easily pass through them into the proximal convoluted tubule. Bowman's capsule. The podocytes forms the inner wall of the Bowman's capsule having gaps of around 25 nanometers wide. These are known as what? Slit pores. They are known as what? Slit pores. Okay. The arrangement or the flow I will tell you in the next coming slide. With the podocytes at forming the inner wall of the Bowman's capsule having gaps about 25 nanometers wide. Okay. The outer wall of the Bowman's capsule consists of unspecialized squamous epithelium, which is flattened. The outer wall or the parietal wall. Proximal convoluted tubule, PCT is made up of simple cuboidal epithelium. Very important, simple cuboidal epithelium. PCT is made up of simple cuboidal epithelium. The Bowman's capsule leads into the proximal convoluted tubule that is lined by a simple cuboidal epithelium bearing a brush border. Very important, bearing a brush border. So it has a microvilli, so it is also known as brush border epithelium. Known as brush border epithelium. Okay. These microvilli are finger-like projections at free end, which increases the surface area for reabsorption. Okay. The cells have numerous mitochondria near the basolateral surface, which allows the reabsorption of salts by active transport. Okay. So they have numerous mitochondria at basolateral surface. At basolateral surface for active reabsorption. Now, one thing I have to show, I'm showing you basically the functional structure of glomerulus, okay, glomerular membrane, and the structure of podocyte. These are podocytes, okay. This is the endothelium of glomerulus, which is fenestrated, having pores, okay. This is the endothelium, which is having pores, okay. So the blood plasma can easily enter through these pores. These pores are known as what? Fenestrae, okay. Now this is the basement membrane of glomerulus, okay. Now, the basement membrane, after it followed by epithelium of visceral layer, that is podocyte layer, that is epithelium. So three layers you can say. Layer of podocytes. Okay, understood. Now, if you will have a look at the structure of podocyte, it will look like this. This is nucleus, right? And this is the cytoplasm. These are the finger-like processes known as pedicels. Understood? So this is the structure of podocyte and the glomerular membrane. Okay. Now we come to loop of Henle. Okay. So PCT is basically made up of simple cuboidal epithelium, and it has microvilli, so it is also known as brush border epithelium. For PCT as well, I am having a picture. Okay. I will make it for you. Actually, I don't have, you know, the diagrams. We were always making on the, hard copy. So I had a habit of that only. But I will try to make it for you. This is the basolateral side having uh, mitochondria. This is the side of basement membrane. Okay, this is nucleus. These are this is the free end having microvilli. Okay, brush border. Okay, they have high enzymatic processes, enzymatic metabolic reactions goes on. That is why they should have good amount of enzyme. That is why they have, you know, Golgi bodies as well. Okay. So this is the proximal convoluted tubule. You can see in the closet. Okay. So my dear students, yeah, today we have actually made good amount of diagrams. But making of diagram gives you a picture in the brain. Okay, the once you make a diagram by yourself, okay, you don't have to go through the text again and again. Okay, so we will be starting loop of Henle. I was thinking we will be starting the loop of Henle in the next turn. Okay, so this is when we have completed the Bowman's capsule, glomeruli. Okay, types of nephrons we have completed. So we will be starting with loop of Henle in the next turn. Okay, till then, bye bye. Take care. Stay safe. Stay healthy.